And once again, welcome to the Los Angeles Birders for December 9th. We are very pleased to have Teresa Pegan with us tonight. She will be presenting a fascinating webinar on the journey and the destination, how seasonal migration leaves a mark on the genomes of birds. And to introduce Teresa, we have Tori Fay, our program chair? I guess so. Program chair to introduce Teresa.
Great.
Tori?
Thanks, Ron. I'm really excited to welcome Dr. Teresa Pegan, a rising star in the world of avian evolutionary biology. This research brings together fieldwork, genomics, and big questions about how birds move through the world and how these movements leave a lasting mark on their DNA. Dr. Pegan first fell in love with migration as a student at Cornell, where the rhythms of bird movement sparked her curiosity about how behaviors like long distance travel shape evolution over time. She went on to earn her PhD at the University of Michigan, where she studied the relationship between migration strategies and genetic diversity, asking questions like, how do migratory birds remain genetically connected across vast distances? What happens in the genome when birds trade stability for seasonal travel? Now a postdoctoral researcher jointly appointed at Harvard and UMass Amherst, Teresa continues to explore how migration, dispersal, and population structure shape the genetic signature of bird species. Her work, which utilizes full genome sequencing and museum specimens, has already begun to reshape our understanding of the evolutionary consequences of movement. In addition to her research, Teresa is a dedicated mentor and collaborator who works across disciplines and institutions. And in 2025, she was recognized as the James G. Cooper Early Professional Award by the American Ornithological Society. Tonight, she'll guide us through how migratory journeys leave deep, traceable imprints in the genome. Please join me in welcoming Dr. Teresa Pegan. And Teresa, I'm going to go ahead and stop sharing my screen so you can start.
All right, yeah, thank you so much for that introduction. I'm going to go ahead and share.
Fantastic.
All right, here we go. Yeah.
There you go. Perfect.
Again, thanks so much for the really kind introduction, and I'm excited to share my research with you all today. Just make sure that I can advance the slide. Okay, here we go. Yeah, so I will give a little bit more background about me and where I am in my career. I'm a postdoctoral fellow, and as Tori indicated, I'm really interested in studying bird migration and genomics. And I kind of got my start thinking about this even before, before I was in college, just because I am also a birder. And I think, as many of us do, I really enjoy going outside and seeing a great diversity of birds all in one place and thinking about the ways that they're similar to each other. Birds that occupy the same habitats often have some similarities, but also the ways in which they're different. And I'm really lucky to have been able to sort of make thinking about these issues of similarities and differences in birds into my career as a scientist. And just a little bit about the sort of journey that I've taken. This is repeating a little bit of what Tori said, but I did my dissertation at the University of Michigan. And most of what I'll share with you today arises from that work. So this is also a bit of a pre-acknowledgement slide. My advisor there was Dr. Ben Winger, and he was a collaborator on all this research. So now I'm a postdoctoral fellow, which means that I have gotten my PhD, and this is like a temporary research focused position before getting into a more permanent position that many folks in science go through. And my particular postdoctoral fellowship is sponsored by the National Science Foundation. And I'm supported by two different institutions, the University of Massachusetts at Amherst and Harvard. And the work that I'm doing now is related to what I'll share today, but it's mostly not completed yet. So that will have to stay for a different time. And then will be starting my own lab next year in August at the University of Illinois Urbana-Champaign, which I'm really excited about. So that's kind of who I am and where I am in my career. And from there, I'm going to go ahead and jump into my presentation. So, just as a little bit of background, oftentimes when we think about evolution, we think about the ways that animals adapt to their environments. For example, this ptarmigan, that's this great white color to help it blend in with the snow, or this loon, which has this great sort of torpedo shape that helps it move aerodynamically through the water. And these are these two bird species have very different habitats. So you can see that they're very suited to the particular sort of habitat and lifestyle that they have. Um, but it's also sometimes the case that we can see organisms that are in the same kind of habitat adapting to it in very different ways. For example, at high latitudes, when the winter comes, it's very challenging to survive. And animals have a wide variety of ways of dealing with this, ranging from things like hibernation, which bats do, food caching, as you know, red squirrels and many bird species also engage in this kind of behavior. But then, of course, the vast majority of birds deal with the challenges of winter by migrating away. And I really like to think about, you know, what drives these different species to end up with different strategies. So today, the group of species that I want to focus on is this group of boreal songbirds and woodpeckers that they occupy the boreal forests of North America, which are shown here in the green color in this map. And they are all broadly co-distributed in this region. If you go out birding in the boreal forest, you will see many of the species that are shown here in this circle. You know, sometimes you can hear them all singing at the same time. So they fundamentally share this habitat during the breeding season. But the breeding season is relatively short at high latitudes. And something really remarkable about these boreal species is that they migrate to vastly different places in the winter. So in this plot, I've connected a sort of arbitrary point in the breeding range. It happens to be in Michigan because that's where I did a lot of this work. But so this point in the breeding range where many of these species breed, each of these lines connects that point to the centroid of the wintering range of each species. So these are not actually migratory tracks that I'm showing here, but they're a sort of indication of how far the species shifts its range in the winter. And you can see that there's this tremendous amount of variation, ranging from species that just shift their ranges a little bit while staying within pretty high latitudes, all the way down to species where the entire species goes to South America for the winter, and pretty much everything in between. And so I really like thinking about why. Why is it the case that these birds, which share some fundamental similarities in the breeding season, do such different things in the winter? And this question of like why birds migrate to the places that they do, I would characterize as one of the great ornithological mysteries of our current time. There are lots of different hypotheses about different factors that might drive species to migrate to different places, such as their habitat requirements and things like that. But when it comes down to it, if you think about species that migrate really long distances, oftentimes when I talk with people about this, they'll say like, "Oh, why do they have to go so far? Why don't they just, you know, go to some place that's a little bit closer, a little easier to get to?" And I have a really hard time as a scientist coming up with a satisfying answer to that question sometimes, which I think sort of reflects how much there is for us to learn still about the behavior of these wonderful species. So I'm going to get into a little bit about how I think about why birds migrate to different places today. And I also like to think about what are the consequences. So these are birds that are going to very different places and exposed to all different kinds of habitats and, you know, in the current day and age, different risks. So these are some important reasons why we might need to think about this kind of question. And of course, as the title of my talk implies, today I'm going to primarily focus about thinking about the genomic consequences of seasonal migration and what we can learn by comparing patterns in the genomes of all these birds. So as an outline of the topics that we'll cover today, first I'm going to give you a little bit of background about how I study these questions using a comparative approach. And then I'm also going to talk a little bit about seasonal migration as a life history strategy, which I'll define what that means a little bit more in a moment, but it's essentially how seasonal migration interacts with the process of how species trade off investment in survival versus reproduction. And then finally, I'll get to the meat of my research about the evolutionary consequences of seasonal migration in patterns in avian genomes. So to dive into this background part, it's really interesting to study evolutionary history, but we can't study it using experimentation the way you sort of often think about with science. You know, we can't go out and manipulate a migratory bird population and wait a few million years to see what the result is. But we still want to be able to ask questions and test hypotheses about birds and other species in nature that we can't do experiments on. So how can we test hypotheses in these cases? Well, one really powerful way is to use what's called a comparative approach, where we're essentially comparing patterns across many different species to look for correlations and trends. And by doing this, we can identify what you could think of as like a natural experiment. So in experimental biology, you might do something over and over again in many experimental replicates and see if the outcome is the same, or to see if you change a certain thing, whether that affects the result. And using the comparative method, we can think of species almost as like replicates, and we can test patterns that way. But of course, it's very important to recognize that species are not completely independent replicates the way we would ideally like for an experiment. And so comparative analyses are also done using methods that account for the phylogenetic history of the species and how they are, some species are more closely related to others than others. And there's a long history of comparative biology in natural history museums. You can imagine sort of pulling out a museum tray and seeing a great diversity of species and comparing their colors or their morphology. And these days, there's also an increasing interest in doing these kinds of comparative studies using other types of data, such as spatial and ecological data, and then, of course, genetic data, which is my main interest at the moment. So, applying the comparative approach has revealed some really intriguing patterns across the tree of life. So, for example, if I show you pictures of two different species and ask you to think about which one you would guess has a longer lifespan. In this case, I'm showing like a tree and a little weed, and you probably have an intuitive sense that organisms that are large, such as a tree, or in the case of mammals, like an orangutan versus a mouse, or in fishes, a shark versus a minnow, that the larger species tend to have longer lives, and smaller species tend to have shorter lives. So this is a general sort of rule across the tree of life, and it's a pattern that arises when we compare lots of species at once. And then, to take a step further, if I asked you to think about which of these different species in these little pairs have more seeds or more babies per year, you might have a sense that, an orangutan has very few offspring every couple years, whereas a mouse can reproduce prolifically. And that's because these animals are sort of falling out on what we call the slow-fast life history continuum, which is this pattern that exists across a lot of different organisms. And the slow-fast life history continuum, it basically reflects the different ways that different species have balanced their limited time and limited energy into survival and reproduction. Because the idea is that if you have a limited budget of time and energy in your life, you can't maximize survival and reproduction at the same time. So on the slow end of the slow-fast life history continuum, we have species that tend to be larger, longer lived, and have fewer offspring per year. And those we call these slow species. We think of them as placing more priority on adult survival, where adults will try to survive to try breeding again, as opposed to just trying to go all out with reproduction in a given year. Whereas on the fast end of this continuum, we have the opposite. The priority is to reproduce as much as possible as soon as possible, and perhaps because survival is less certain. So that was a bit of a tangent to sort of think about what we can learn with the comparative method. But I do want to bring it back to this question about species sort of reacting to their environment in different ways. And it's often the case that when you have a group of species that share a particular habitat and they deal with the challenges of it in different ways, those differences can be related to differences in life history strategy, to the extent to which they're prioritizing survival versus reproduction. So, how does migration distance fit into this slow, fast life history continuum? And do these different migratory strategies that these different birds use, do they reflect the slow, fast life history continuum? Well, from one perspective, we might think of migration as being very risky to survival, and therefore an element of the sort of fast end of this continuum. And migration is risky, and I think one of the most evocative examples of this that I've seen recently is this in this paper that came out a couple years ago. And it was a paper about the diets of tiger sharks. And they found that during the migratory season, the spring and the fall, tiger sharks had a huge proportion of the tiger sharks that they assessed the diet of by making the shark barf into this bucket actually had the remains of songbirds and woodpeckers in their stomachs. And of course, sharks are not going out and hunting migratory birds. So what this reflects is the fact that so many migratory birds perish as they try to migrate across the ocean, and these sharks are scavenging their remains. So migration is very risky to survival, and maybe therefore something that we think of as being associated with the fast end of the slow, fast life history continuum. But it's also really important to think about when we think about birds choosing to make migratory journeys, what the alternative to that would be. And for birds that breed at really high latitudes, the alternative to migration is trying to stick it out during the winter when the temperatures are very cold and resources may be limited. And furthermore, when you adapt to deal with winter by departing from the breeding grounds, that could be potentially seen as a sacrifice to your ability to maximize reproductive output. For one thing, there's a tremendous pressure on birds to get to their breeding grounds and start trying to attract a mate as soon as possible. And so, the further away you go, the more difficult it is to get back in a timely fashion. And then also, there could be sacrifices for birds that leave the breeding grounds before the weather gets really bad. They may be sort of sacrificing opportunities to extend their breeding season. And to think about that sort of time limit imposed by migration in a little bit more detail, one of the projects I did at Michigan involved using citizen science data from eBird to look at the length of the breeding season of these 35 boreal bird species shown in this circle here. And it turns out that long distance migrants tend to arrive in the boreal region late and depart from it early. And if you've ever been birding during the migration season, you might have also sort of noticed this. We tend to get short distance migrants coming through early and long distance migrants coming through later. And then in the fall, it's the opposite, where the long distance migrants are coming through first and the short distance migrants come through later. And in the boreal region, the species that spend the most time on their breeding ranges are spending almost like twice as much time in those areas as the really long distance migrants that are arriving later and departing earlier. So this really does indicate that long distance migrants are making a time sacrifice, at least, for their migratory journeys. So we wanted to see whether this sacrifice of time on the breeding grounds was accompanied by sacrifice of reproductive opportunity. And to do that, we dug into this great data set that's managed by the Institute of Bird Populations. And from these sort of standardized mist netting protocols from a program called MAPS. We often think of mist netting as something that especially happens during migration because we want to see where birds go. But these MAPS mist netting efforts are looking at birds during the breeding season, and because of that, they're able to make estimates of annual survival and reproductive output rates for different bird species across North America. So we first looked at, in boreal bird species, whether there was a relationship between migration distance of a species and their annual fecundity or their reproductive output. And so I'm showing again this picture depicting the lengths of their breeding seasons, where the longer distance migrants, which are in these redder colors, have shorter breeding seasons. And we found that it is also indeed the case that long distance migrants tend to have lower annual fecundity than short distance migrants. So you can see this negative trend in the scatter plot, where the x-axis is the distance a species migrates, the y-axis is a sort of standardized metric of its annual fecundity, and each point represents a species. And we also found that this is driven in a large part by the fact that short distance migrants are often able to have two nests in a given breeding season, and long distance migrants don't have that opportunity because they don't have enough time. So there really is, when you're a long distance migrant and you're leaving the breeding grounds a little earlier, you really are sacrificing an attempt maybe to have a whole other nest compared to species that choose to migrate shorter distances. So if migration involves this sort of sacrifice of reproductive opportunity, the question is then, is it balanced by a benefit to survival, as we would expect, based on this hypothesis about life history? And the answer is actually yes. So perhaps counterintuitively, species that migrate the longest distances tend to have fairly high annual adult survival compared to those that migrate shorter distances. So even though they are undertaking these longer journeys that we might think of as being more risky, they actually tend to survive as adults at higher rates. And so to return to this question about why different boreal bird species migrate to different places, again, it's a very complex and multifaceted question, but I would suggest that at least sort of one facet of why is that they have different life history strategies, and something about these different migratory distances reflect the different ways that these species have of balancing their investment in survival versus reproduction, where migration is actually fundamentally, even though it's thought of as being risky, it's fundamentally a survival adaptation, where birds are making these incredible journeys in order to better survive this very harsh, difficult winter season at high latitudes. And it may have something to do with where that sort of survival takes place. So here I'm showing an example of two related thrush species. They're in the same genus. On the left is the hermit thrush, which is a relatively short distance migrant. And even though they do migrate south for the winter, as you can see in this picture, a lot of them are often spending the North American winter in areas that are still going to be challenged by cold and resource limitation, and in fact, even sometimes snowstorms, as seen in the picture. And by contrast, the bird that's shown on the right is a Swainson's thrush, and these are really long distance migrants that are all going down to South America for the winter. And there, they experience warmer climates and probably more abundant resources. And so that may be why they're able to sort of have higher rates of survival. Okay, so with all this in the background, what are the evolutionary consequences of migration in the avian genome? So sometimes patterns in the genome are thought of as being like a blueprint for the organism, although I would argue this is quite a strong oversimplification. But nonetheless, it is the case that sometimes you can find a little pattern that exists in the DNA of a species and relate that to something that the species does or some aspect of its biology. So there are certain genes in the genome that play a role in how birds migrate, for example. But another really interesting aspect of DNA is that because it's inherited across generations, patterns in the genome also reflect processes that have occurred in the history of a population. This is just a little picture of a pedigree. You can think of it even at that sort of very fine scale, like the number of offspring that a couple has reflects something to do with the history of that particular family. And these patterns that reflect these historical processes are particularly evident when we compare genomes from many individuals that are closely related to each other. And this is a sort of subset of genomics that we call population genetics. So there are two long-standing hypotheses in population genetics that I want to talk about today that have to do with how events in evolutionary history shape patterns in the genome. The first is the idea that spatial patterns in genetic relatedness, so patterns in how closely related individuals are compared to how close they are in geographic space, are thought to reflect the history of movement in that population. Where when individuals move around more and are sort of mixing through breeding, there's high gene flow that sort of erases spatial patterns. Whereas if they are not moving around as much, then you get more spatial patterns in the genome. And then the second hypothesis I want to talk about is about the amount of genetic diversity in populations, which are thought to at least in part reflect the history of the size of the population, where larger populations tend to have more genetic diversity, and also the demographic stability of the population, or how stable that size has been through time and the extent to which it's gone through fluctuations. And both of these are processes that I would argue potentially interact with seasonal migration. It's really intuitive to think that migratory movements, which are, of course, tremendous movements in and of themselves, might interact with the movements that individuals have within the population. And then, of course, as I've just described, migration, through its effect on life history, potentially interacts with processes that influence population size and stability. So, what we're going to spend the rest of today talking about is how migration distance of these different species co-varies with the amount and spatial distribution of genetic diversity in these populations. So to introduce you to this idea of spatial structure, this is related to hypotheses about how movement behavior, the history of movement in a population, influences spatial patterns. We say that a population shows spatial structure when individuals that are close to each other in geographic space are also closely related. And I've made this little cartoon to sort of illustrate this here. You can see a map of the boreal region with some sampling locations on the lower part of the image. And on the upper part, I'm showing something called a PCA. That's made with genetic information. And the only thing you really need to know about a PCA is that it's a way of sort of taking a bunch of really multivariate information from the genome and summarizing it in 2 dimensions. And in this case, you can see that even though in the PCA we didn't feed it any information about the location where the samples came from, the samples that are close to each other in geographic space that share the same color also tend to end up close to each other in genetic space in the PCA. And this is reflecting the fact that individuals close in space are more closely related to each other than a pair of individuals. If you took a pair of individuals from disparate locations, those would be less related. So this is a species that shows spatial structure. And gene flow, which is a term used for the sort of movements of genes that take place when individuals from different places sort of move around and interbreed with each other. Gene flow disrupts this pattern. So if there's a lot of gene flow, you would not see a pattern like this because individuals that are far from each other in geographic space might be closely related in genetic space. It might just be that one of their relatives sort of went on a long distance dispersal. And it's intuitive to predict that migration, seasonal migration, might interact with this process, even though it's important to remember that migration itself, in the avian sense, is a round trip journey. Birds migrate south for the winter and then they come back. They're not doing any interbreeding on their non-breeding range, and the process of migration is not exactly synonymous with the process of dispersal. Dispersal is a movement from an individual from one breeding location to another, or from where it was born to where it eventually breeds. And dispersal is the behavior that mediates gene flow. Now, birds that can migrate super long distances, it's really easy to imagine that they can also disperse long distances. This is a picture of a track, a migratory track that was measured in this Connecticut warbler, which is a boreal species. And you can see that it was able to travel an incredible distance on migration and even cross the ocean in only a couple of days. So if you're able to do that kind of travel during migration, then you'd think it would be no problem for that individual to move, a long distance across the breeding range if it wanted to. So potentially we could think of migration as something that promotes gene flow in space. But migratory journeys are also characterized by another sort of really important feature, which we call phylopatry. And phylopatry refers to the tendency of migratory birds to return to the same places over and over again. Phylopatry is just like Greek for love of homeland. It's kind of a silly jargon word, but it is remarkably the case that migratory birds come back to the same places over and over again, even to the extent of coming back to the exact same, bush or tree that they nested in before. And this picture of this migratory track actually sort of also reflects migratory phylopatry, because on small species like this Connecticut warbler, we can't put tags for tracking on migration that can transmit to satellites. Those are too big for them to carry. So instead, we have to use these tiny little like geolocator tags that collect the data but don't transmit it. And the only way to get this kind of track back is to recapture the exact same individual again. And the prospect of finding a bird, in the entire world after you've tagged it, the following year would be pretty much impossible, and is only made possible by the fact that you can go back to the same place and expect that bird to return. So that there a lot of tracking studies really have relied on this strong phylopatry. And so this means that migration is characterized by these two conflicting features: mobility that might promote dispersal versus phylopatry of birds returning to the same places over and over again. That's kind of the opposite of dispersal. So I wanted to know how these conflicting features of migration play out and influence gene flow over evolutionary time. So here's some predictions about what that relationship might look like. If we think that migratory mobility promotes gene flow, we might expect to see a positive relationship between the distance a species migrates and the amount of gene flow that exists in its breeding range, where gene flow is kind of the thing that erases spatial structure. Or if we think that migratory phylopatry is more important and that maybe it's stronger in the longer distance migrants that are traveling further just to get back to the same places, we might see the opposite relationship. Or maybe if these two factors sort of balance out against each other, we might consider that we'll see no relationship. So in order to test these predictions, I generated a really big genomic data set from a really large collection of samples from across the boreal breeding range of 35 species. The sampling locations are shown here in this map. And the genomic resources came from a lot of different institutions, especially natural history museums, as well as a lot of field work from myself and my collaborators over the years. And in order to take those genomic resources and turn them into analyzable sequence data, I again used these 35 species. I ended up with about 1,700 samples from across those different species. And I used a technique called low coverage whole genome sequencing. The details of this don't matter too much, but we're getting whole genome sequences from these birds, and using special kinds of analyses that account for a little bit of uncertainty in some parts of the genome. And I used PCA like I showed you before, to assess spatial structure. So beyond making, this is again a PCA plot. This one happens to be from blue-headed vireo, which is a species that shows spatial structure. But beyond these kinds of plots, which are kind of qualitative, we can also quantify spatial structure with what we call a slope of isolation by distance. And this is what it looks like in a scatter plot form. We've just kind of taken pairs of individuals and plotted on the x-axis the geographic distance between them, and on the y-axis a value called genetic covariance. And genetic covariance tends to be positive when individuals are closely related and negative when they're more distantly related. And so when there's this nice spatial pattern like this, we see a negative slope, where the individuals that are far in space tend to have negative covariance, and those that are close in space tend to have positive covariance. So I made these kinds of plots and I estimated these slopes of isolation by distance for all of the different 35 species in my study. And I found a lot of variation. There were some cases like the blue-headed vireo and this mourning warbler where there are really clear spatial patterns, which means that there is not enough rampant gene flow across the breeding range to erase those patterns. Whereas there were other cases, such as this Tennessee warbler and the white-throated sparrow, where individuals were all, genomically, they were all mixed up in space. You couldn't really predict how closely related a pair of individuals were based on where they were in geographic space. So then the question is, can migratory behavior explain all of this variation between species that show structure and those that don't? Well, the first thing I compared was between species that migrate and species that don't. And there aren't very many species in the boreal region that don't migrate for the winter, but you can still nonetheless clearly see that non-migratory species tend to have a more negative isolation by distance slope, which means that they show more spatial structure than migratory birds. And this kind of aligns with what we know about how migration interacts with dispersal. We know that migratory birds do tend to disperse more than non-migratory birds. So they do have overall a bit more gene flow across their ranges than non-migratory species. However, if we look within the species that do migrate, which is most of them, and we look at the relationship between migration distance and that slope of isolation by distance, we see that there isn't really so much of a relationship here. There's no statistically significant relationship in this particular plot. And what's going on here is that the species that migrate very long distances, which are shown in the redder colors, some of them show spatial structure with a really negative isolation by distance slope, and some of them don't. And likewise, that's also the case with shorter distance migrants. So whether or not a species migrates a long distance doesn't really tell you much about, it doesn't really allow you to predict whether they show a lot of gene flow in their breeding range or not, which is really interesting. And I think it reflects the fact that gene flow and mobility are not necessarily connected in migratory birds. In a lot of species that don't migrate, how well they're able to move around does really strongly predict how much dispersal there is. But in migratory birds, there must be other things that are playing a more important role. And this is really like a consequence when migratory species have these spatial patterns. This is a consequence not of it's not because they can't move around very much, it's because they're choosing not to move around much in between their breeding seasons. They're showing this migratory phylopatry. And I think this is really remarkable when we think about it in a spatial sense. This is the range of this mourning warbler shown on the right here. And you can see in the PCA plot that there is spatial structure where these colors are separating out. And we would not see that pattern if there was just a little bit more movement of individuals between breeding areas, a little bit more dispersal in the breeding range. They're not undergoing that kind of movement. And that's really remarkable given that the scale of the migratory movements that the entire population makes every year are like pretty much the same distance as the entire breadth of the breeding range. So here we see this really strong signature of migratory phylopatry in these population genomic patterns. Okay, the last thing I want to talk about is Um, Now moving from thinking about how genetic diversity is arranged in space to thinking about how much genetic diversity there is in a population. And there's this model in biology called a population bottleneck, where when a population size crashes, we know that that often causes a very long-term reduction of genetic diversity. Even if the population size is able to sort of recover relatively quickly, the population will experience low genetic diversity for a long time. And so the question is, does migration distance influence these kinds of demographic processes that shape the genetic diversity of different species? And well, if we think back to the section about life history, it's one possible hypothesis we could come up with is that long distance migration helps buffer populations against sort of population size fluctuations. For example, if you think of a short distance migrant that has to spend the entire annual cycle within north temperate latitudes, there could be some winters where a really strong storm comes or it's particularly cold that wipes out a lot of individuals in the population and causes sort of an erosion of genetic diversity. Whereas species where all of the individuals are going this really long distance to more stable and resource-rich habitats, maybe they see a sort of stabler population over time. So that's one hypothesis, and under this hypothesis, we might predict a positive relationship between the distance a species travels on migration and how much genetic diversity there is in that species. But it's also valid to hypothesize the opposite, thinking back to these sharks that are scavenging all these migratory birds. Migration itself, of course, also exposes populations to risk. You could get a hurricane out over the ocean while a large number of individuals are trying to migrate. And so if we think of migration as more of a driver of instability in population size, we might predict a negative relationship between the distance a species travels and how much genetic diversity there is in the population. So I used the same genomic data set from 35 boreal species that I showed before in the spatial section to test this hypothesis as well, looking at just the amount of diversity there is in populations. And I was actually very surprised to find this result, to find such a strong relationship here between the distance that a species migrates and how much genetic diversity there is in its boreal population. This is a strong positive relationship where these long distance migrating species that are shown in red have higher values of genetic diversity than short distance migrants, and correspondingly, the short distance migrants tend to have more genetic diversity than non-migrants, which I'm showing here in gray. And this pattern exists at multiple different levels, so we can think about it in terms of just how many genetic variants there are if we look across all individuals. But it even exists down to the level of heterozygosity within individuals. And heterozygosity is this just a metric of you have two copies of the genome, and whether the two bases at a particular genomic locus are the same or not. Long distance migrants tend to have more variety within their own genome than individuals from short migrating species. So why is this the case? Well, I set up this hypothesis about migration potentially helping buffer populations against instability and fluctuations in population size. And I think it's really interesting to observe that if you think about differences in migration distance while holding the breeding range constant, as I have done here in this study, then the distance a species migrates is also a reflection of the latitude at which it spends the non-breeding portion of the year. And when we think about it from that perspective, this genetic diversity pattern has this neat parallel with a pattern that has been described across lots of different species, including many non-migratory species. In the middle here, I'm showing a figure from a paper that came out a few years ago that looked at genetic diversity across almost 7,000 vertebrate species. And they have this like histogram that's on its side here. But what you can notice is that the values of these bars in red that are in tropical regions tend to be higher than the values nearer to the poles. And in this paper, they hypothesize that this is because tropical environments have experienced more stable climates over time. And so this is potentially something that plays out even in birds that are only spending part of the year in the tropics versus spending the entire year in the temperate zone. So I think it's really interesting. I sort of started this project thinking a lot about how migratory movements potentially influence genomic patterns in birds. And I found that there wasn't really a strong pattern there, but that this main genomic signature associated with migration distance in boreal birds has more to do with the amount of genetic diversity that they show, and that might be related to where they're spending the non-breeding season. So, pardon this slightly cheesy analogy that I'm making, but perhaps in migratory birds, when we think of the population genetic consequences of seasonal migration, maybe the destination actually matters more than the movements of the journey itself. And this is what I just said, just to reiterate. So long distance migration, it doesn't really promote gene flow, but it is associated with signatures of population stability that might be related with what environments they encounter during the non-breeding period. But I think it's really important to point out that even if long distance migrants show this genomic signature that suggests they've had long-term population stability, that this really strongly contrasts with present-day instability experienced by migratory populations. A large proportion of migratory birds are in decline, and long-distance migrants are some of the most endangered of migratory species. And on the one hand, my finding that in the boreal system, at least, long distance migrants tend to have high genetic diversity might be cause for optimism because we think that high genetic diversity is good for adaptation. It allows there to be a lot of different variants around that evolution might be able to work with to help the population sort of adapt to new conditions. But it's also really important to recognize that this pattern suggests that the scale of declines we're experiencing now may have little precedent, at least in the recent evolutionary history of these species. These are not species, it seems, that are sort of used to going through big fluctuations in population size. And so that may make it more difficult for them to recover from population decline. So, with that, I want to thank you all for listening, and thank you to the many institutions that have supported me over the years and my research financially with grants, and as well as all of the institutions that provided the genomic resources that I used in my analyses. And with that, I will be happy to take any questions.
Fantastic. If you have any questions, please pop them into the Q&A section or tab down at the bottom of your screen. That helps us kind of keep track of everything. And Teresa, that was wonderful. I loved it.
Yeah, thank you so much.
Great.
And let's see here.
I'll
Yeah,
go ahead
you
and
can,
start with the question if that's okay.
yeah, go ahead.
Yeah, go for it, Tori.
So this was really fascinating. I loved all of it. I was kind of wondering as you were talking about this, just about a local bird that we have, the Allen's hummingbird, which has two subspecies, one which is migratory, one which is not. And I wonder what you think you might find if you looked at their genomics.
Yeah, that's a great question. And that's a species that's the one that sort of has been expanding a little bit too, right?
Yeah, the non-migratory one is, I believe, is the one that's been expanding.
Yes, I believe
okay,
so.
yeah. Yeah, well, one thing that I think is really important about the sort of the patterns that I found with migration distance is I do think it makes a difference whether you hold the breeding range constant or not. So if we were to look at a species where some individuals migrate and some don't, but they all come back and breed in the same place, then I suspect we would see something similar where the migratory individuals are maybe having lower reproductive output in a given year and sort of investing more in survival. But in cases where there are species that have a non-breeding, a non-migratory population that's maybe further south and a migratory population further north, I'm not as sure about what would happen with the genetic diversity there because we tend to see higher genetic diversity the further south you go. And I think there's a big difference between being a non-migratory population in a sort of warm latitude versus being non-migratory in the boreal region where you're really facing these sort of severe challenges and potentially incidents that cause large amounts of mortality. So that would be that. I often chat with people when I share my research about what happens within partial migrants, which are those species where some migrate and some don't. And I think that's a really interesting question to pursue, but I haven't actually done any analyses like that before. So yeah, thanks for the question.
Thank you.
Lily has a question on the Q&A. Can certain migratory groups, depending on distance and genomics, be more vulnerable to climate change?
Yeah, for sure. I think this is like $1 million question in bird conservation is like how do we predict which species are going to have the most challenges and how do we protect them? And yeah, well, from one perspective, long distance migrants are challenging to conserve because we need to help conserve habitat across such different vast spaces, right? If you've got a non-migratory species, you know where it is, and it's like, okay, this is what we have to protect. But long distance migrants, it's so much more complicated. And then the thing that's really challenging for migratory species, in part, is adjusting the timeline of their annual cycle. As spring gets earlier, the spring green up gets earlier every year, some bird species are able to kind of track that and come back earlier, and some are not. And we think that that long distance migratory species seem to show particular um, inability, or they're a little bit less good at tracking the spring green up than short distance migrants, which might be partly because a short distance migrant that's in North America can sort of sense that spring is coming and be like, "Oh, I better get going." Whereas if you're in South America, it's like whatever you're experiencing down there isn't really related to what's going on in the breeding range.
Right.
So that's definitely a challenge. And again, there's this idea that genetic diversity helps populations adapt to new conditions, but it's still very unclear how that works in any particular case. And I think it's unclear if it's really relevant to whether migrants can adapt to new conditions or not. That's something that I'm really interested in studying in my current research.
Cool.
Right. Question from Carol, which is very similar with short distance migrants having less genetic variability. Does that make them more vulnerable to climate change?
Yeah. Again, great question. And there's this prediction out there that there have been some studies that have sort of directly looked at the relationship between genetic diversity in populations and how rapidly they can adapt to new conditions. And from that perspective, we might think that yes, populations with less diversity might face more challenges doing that. But there's so much nuance and complexity that in any given case, I think it requires some dedicated study to find out what's really going on. And for example, another thing that is not really clear is um, one way for a population to sort of react to changing conditions is through evolution, which involves changes in the actual genetic variants in the population over time. But another way is through flexibility. Individuals that can, with the same genetic makeup, even within their own lifetimes, that can start doing things a little bit differently. And it's a really big question how flexible different migratory populations are and whether that, I think that there's a sense that maybe long distance migrants are less flexible than short distance migrants. And so from that perspective, short distance migrants might have a bit of an advantage when it comes to changing the timing of their migration, for example. But these are all still really important questions that are the topic of active study in birds and other organisms. So yeah, there's a lot more to learn.
Oh, great. Thank you. Scott has an interesting question. Do eruptive birds like boreal finches have more diversity than non-eruptive boreal species?
Yeah, that's a great question. And you may have noticed I didn't have any eruptive finches or nuthatches in my study. And that was purposeful because we began this study primarily thinking about spatial patterns. And we thought that, I haven't actually studied them, so I can't guarantee that this is true, but I think it's pretty likely that eruptive species like finches, where the population is moving around a lot to breed, not just to migrate, where they really are, popping up and breeding in one place and then going somewhere else to breed in the next year, those species probably don't have spatial patterns. And
Hmm.
so that's why we didn't include them in the study because we were like, we have it costs a lot of money to sequence genomes, so we wanted to focus on species that were more traditionally migratory, where we could measure a migration distance. Um, and it's a really good question about how those sort of eruptive dynamics influence genetic diversity. I think the answer partly depends on whether these species go through big population size fluctuations or not, and I don't actually know the answer to that. Maybe there's some folks who study finches who do, but yeah, in insects, it's thought that species that can sort of really rapidly proliferate, like certain kinds of, there's a lot of insects where they can have like these huge outbreaks. Those dynamics, I think, are thought to potentially rate they allow populations to have higher genetic diversity because the population gets really big. But I'm not sure that I sort of suspect that if that's happening in birds like finches, that it's sort of at a much lower degree than in insects. So I don't know. That's a great question. That'd be a neat thing to study.
Great. Thank you. Mike asked, are there similar studies with similar results or corollaries for raptors to what you found for your study?
Yeah, good question. I don't know of any if there are, but yeah, I was specifically focusing on these very small birds, and I think that you could look at these patterns in lots of other species as well, and you might find similar things or different things. One thing that's for sure is if you compare a raptor species with a songbird species, there are a lot of reasons why levels of genetic diversity might be different. If you think back to earlier in my presentation, again, bigger species tend to have different genetic diversity patterns than smaller species. And so, in my case, by focusing on small birds, I was trying to sort of control for that at least a little bit. Um, but you might be able to see something similar in parallel if you looked at a bunch of raptor populations. That would be neat to do, but I don't know of any comparative raptor population genetic studies in North America, but it would be a great thing to study.
Mhm. Great. Um, and we have a question from the YouTube live stream from Alexander. He says, great talk. May have missed this before, but do you have any species in your data set that showed signs of past bottleneck events?
Hmm.
Yeah, great question. So there's been a sort of paradigm in thinking about the evolutionary history of species in North America that glacial cycles caused widespread bottlenecks, right? And I did a related study on some of these birds and found that it wasn't that simple. That a lot of a lot of populations of boreal birds seem to have been expanding since longer ago than the last glacial maximum. And so I do think that glacial cycles across, the hundreds of thousands to million years of Pleistocene glacial history, I do think that they were disruptive to populations and may have caused population size changes over time. But I don't think that there's evidence that there was a very recent bottleneck in these boreal populations. And even though the glaciers only about 20,000 years ago were pretty much covering the area where the boreal forest is now, um, it could be the case that there was plenty of suitable habitat down along the edge of the glacier. So, um,
Hmm.
so I don't see any evidence of like a really severe recent bottleneck in any of these species, but I do wonder whether the differences in genetic diversity can be explained by processes that are occurring sort of at less extreme scales, but maybe more often. over evolutionary time. So, instead of like a major bottleneck reducing the population to something tiny, just thinking about reductions in population size that happen recurrently as the north temperate wintering species are exposed to harsher conditions. But that's just a hypothesis. There's still lots to study with these patterns.
I
Well,
had a
good.
quick question, Teresa. I was
Yeah.
wondering how, like, all your study species were in the boreal forest and like eastern migrants. Do you expect to see like similar patterns play out in like with like western birds with like the mountains and like the more arid climates and stuff?
Yeah, I think that if you took the community of birds on the other side of the Rockies and you did something similar where you look at species that are breeding in the same place but migrating different distances, I would, perhaps I'm biased, but I would predict that you would see something similar. Um, Again, if you compared the northern breeding populations with populations that are breeding further south in the Rockies, would you see something similar, or would it be would that latitudinal pattern and genetic diversity kind of start to be more important? I'm not sure. Um, I species and there are a lot of sort of pairs of species where one is in the east and one is in the west, or maybe there's pairs of subspecies around the Rockies. And I did exclude those from my study on spatial patterns because I was interested in how migration influences movements that aren't really affected by something like a big mountain range in the way. But I do expect that those, oftentimes those populations are a little bit diverged from the Eastern populations.
Awesome, thank you.
Yeah.
Great.
Any more
And
questions?
I
Q&A
don't
is
see
empty at
any.
the moment.
I don't see anything else. Teresa, thank you very much for a wonderful webinar tonight. I think we all enjoy that. And we're getting lots of thank yous on the chat. So a lot of people join me in that.
Well, yeah,
Um,
thank you for having me.
Thanks you again. Thank you again. Please join us for next month's webinar. They always are fascinating, and they get better and better towards the end of the year. Even though Teresa's was wonderful. And with that, we will see everyone next month, if not out in the field. And I want good night, everyone. We'll
Thank you,
take
Teresa.
care.
Okay,
Thank
thank
you
you,
so much.
Thanks.
That
Teresa.
was
Thanks
amazing.
again.
Thank
Good
you.
night,
Good
everyone.
night.
Good night.